day, everyone. I am Miss Borja, your subject teacher in science. I am going to teach you General Biology 1 for this semester. Today, we are going to learn new things and it's gonna be fun. So, come and let's enjoy learning. Hello guys, how are you doing? I hope you are all fine. Today we are going to discuss lesson number 8, biological molecules, enzymes. And of course, for our objectives, first, describe the components of an enzyme. Next, explain the role of enzymes in the body and how does it works, and give examples of some specific enzymes. Okay, so enzymes help speed up chemical reactions in the human body. They bind to molecules and alter them in specific ways. They are essential for respiration, digesting food, muscle and nerve function, among thousands of other roles. In this lesson, we will explain what an enzyme is, how it works, and give some common examples of enzymes in the human body. Enzymes are built of proteins folded into complicated shapes. They are present throughout the body. The chemical reactions that keep us alive, our metabolism, rely on the work that enzymes carry out. Enzymes speed up or catalyze chemical reactions. In some cases, enzymes can make a chemical reaction millions of times faster than it would have been without it. A substrate binds to the active site trusted source of an enzyme and is converted into products. Once the product leaves the active site, the enzyme is ready to attach to a new substrate and repeat the process. So, what do enzymes do? First, in the digestive system, enzymes help the body break down larger complex molecules into smaller molecules such as glucose so that the body can use them as fuel. Another one is for DNA replication. Each cell in your body contains DNA. Each time a cell divides, that DNA needs to be copied. Enzymes help in this process by unwinding the DNA coils and copying the information. Also, for liver enzymes, the liver breaks down toxins in the body. To do this, it uses a range of enzymes. Now, how enzymes work? Enzymes work by lock and key model. The lock and key model was first proposed in 1894. In this model, an enzyme's active site is a specific shape and only the substrate will fit into it, like a lock and key. This model has now been updated and is called the induced fit model. In this model, the active site changes shape as it interacts with the substrate. Once the substrate is fully locked in and in the exact position, the catalysis can begin. Enzymes can only work in certain conditions. Most enzymes in the human body work best at around 37 degrees Celsius body temperature. At lower temperatures, they will still work but much more slowly. Similarly, enzymes can only function in a certain pH range. Their preference depends on where they are found in the body. For instance, enzymes in the intestines work best at 7.5 pH, whereas enzymes in the stomach work best at pH 2 because the stomach is much more acidic. If the temperature is too high or if the environment is too acidic or alkaline, the enzyme changes shape. This alters the shape of the active site so that substrate cannot bind to it. The enzyme has become denatured. Let's talk about the enzyme's inhibition. To ensure that the body system works correctly, sometimes enzymes need to be slowed down. For instance, if an enzyme is making too much of a product, there needs to be a way to reduce or stop production. enzymes activity can be inhibited in a number of ways. We have the reversible and irreversible. For reversible, there are three types. First, competitive inhibitions. In competitive inhibitions, a molecule blocks the active site so that the substrate has to compete with the inhibitor to attach to the enzymes. Next, non-competitive inhibitions. A molecule binds to an enzyme somewhere other than the active site and reduces how effectively it works. And for the third one, the uncompetitive inhibitions, the inhibitor binds to the enzymes and substrate after they have bound to each other. The products leave the active site less easily and the reaction is slowed down. And for the next type, we have the irreversible inhibitions. An irreversible inhibitor binds to an enzyme and permanently inactivates it. Let us proceed to the examples of specific enzymes. There are thousands of enzymes in the human body. Here are just a few examples. First, we have the lipase, a group of enzymes that help digest fats in the gut. Amylase help change starches into sugar. Amylase is found in saliva. Maltase, also found in saliva. It breaks the sugar maltose into glucose. Maltose is found in foods such as potatoes, pasta, and beer. We also have trypsin, found in the small intestine. It breaks protein down into amino acids. We also have the lactase, also found in the small intestine. It breaks lactose, the sugar in milk, into glucose and galactose. We also have the helicase, unravels DNA. We have DNA polymerase, synthesized DNA from deoxyribonucleotides. And we have the acetylcholinesterase. It breaks down the neurotransmitter acetylcholine in nerves and muscles. Okay, so enzymes play a huge part in the day-to-day running of the human body. By binding to altering compounds, they are vital for the proper functioning of the digestive system, nervous system, muscle, and much more. And that's it for our today's lesson. See you again next time, and thank you guys for watching.